All right. I think it's about time for us to get going. I'm Mark Williams. I'm in the Department of Horticulture, and I'm hosting today Dr. Barney Whitman from the University of Georgia. He's in the Department of Microbiology, and the place where we intersected, actually, when I was at the University of Georgia, probably 8, 10 years ago, um, soft money position at the time, was in soil and where plants and soils interact. And Barney was one of the people that had kind of been leading the way in developing some of the methods for studying the soil microbiome, looking at some of the genes that were good markers for describing what he's going to talk about today, the immense soil microbiome. And of course, nowadays we've got an opposite problem. We've got so much data that we don't know what to do with it all. But back then, it was a pretty hardcore churning out of clone libraries and working really hard to get somewhere even close to trying to get some kind of semblance of what the soil microbiome was. And normally, what would happen is you would take a sample and you get 100 and 200, 300 bacterial species, and you knew based upon when you started looking at the data that there was going to be at least 10 times that many. And so you weren't even beginning to describe all of the diversity that was there. But Barney is uniquely qualified to talk to us today because he's kind of been at the forefront of that. He's backed off a little bit from competition from me. He's had to kind of back off a little bit. He also does a lot of work on anaerobic bacteria um, and many other areas. But anyway, I'd like to introduce. Well, one last thing. I'd also like to say, you know, I'm always appreciative of Barney because he was one of those people that, he respected me, of course, for what I could do, but he also wanted to help. He's the kind of person that wants to kind of help other people, and he was glad to work with me. And as a young scientist at the time, not so young now, but as a pretty young scientist, he was the kind of person that wanted to help. And so I thank him for that, and I want to make sure he knows that to be true. So with that, Barney, please take over.
Well, thank you, Mark. Well, it's really a pleasure to be here, and I really enjoy visiting Virginia Tech, and I've worked with a number of people in a number of departments here for several decades, really. So it's really a great place to be. And I would like to say that Mark is a great collaborator because he has so much strength in soil science and plant biology that he could contribute, and none of the studies that we did would have been successful without him. And it's nice to have people who are willing to carry their load plus a little bit of mine when you're working with them. But anyway, today I want to talk about the immense soil microbiome, but I want to talk about it in two ways. One, I want to try to say what it means from the point of view of a microbiologist, and then also kind of say where we are and where we'd like to go. And the major co-conspirators is Mark, of course, who's currently here, and then a man who is a postdoctoral associate in my lab, Kamlesh Janjeed, and he's currently at the Microbial Culture Collection in Pune, India. And these two did most of the experimental work, and I did most of the kibitzing. Other people that were involved, John Blair, Alan Franz Luber, and Thomas Schmidt provided the samples for a lot of the studies we're talking about. And then I've also worked a lot with Charles Chu and Dave Coleman over the years on my other soil-related work. So why study soil? I mean, this is so obvious, but soil is the foundation of civilization, providing essential ecosystem services through agriculture. I don't think we could possibly underestimate the importance of soil to modern life. But to try to put it in perspective, if we look at it's the major reservoir of organic carbon on Earth, with the biomass of land plants about 560 petagrams. or 10 to the 15th grams carbon, and soil carbon is about 3 times that. So if nothing else, there's just a huge amount of soil and soil carbon on Earth. But soil is also an important source or sink for all the major greenhouse gases. So not only is soil land, obviously, but it's also affecting the atmosphere and has a tremendous role in the biochemistry of life. The soil microbiome is vast, and so this is an estimate of microbial biomass based upon the traditional methods of fumigation techniques from Zhu et al. In 2013, and he looked at the top meter of soil and estimated about 23 petagrams of carbon in the living microbial biomass. So this is presumably bacterial and fungal. A number of years ago with Dave Coleman, we estimated the number of bacterial cells by counting in the top 8 meters, and we got about the same amount of carbon. So the numbers are a little bit different, but they're close enough. And if you compare them to terrestrial plants at about 560 petagrams of carbon. So the amount of carbon is much less than in plants, but for plant carbon, it's a little misleading because most of that's structural carbon or wood. And so if you really want to look at the amount of living material, I'd argue that nitrogen is probably a better indicator. So our two estimates for microbial nitrogen in soil are about 4 or 6 petagrams of nitrogen compared to about 10 for plants. So the bottom line then is that the terrestrial living material of microorganisms is probably maybe about half of what we see on plants. Now plants are pretty obvious because we can see them, but this microbial biomass is invisible. So we'd like to see if we can answer just some basic questions about this. And like we'd like to know who is there, who these guys are, what are the structures of the communities, how does these soil communities respond to change, and does vegetation affect the composition of bacteria in soil? And I would say when we started this work, I don't know, maybe 10 or 15 years ago, I would have guessed wrong on answers to all these questions. The things just were not intuitive. They weren't, we actually had to do the experiment to find out what was going on. It wasn't something you could have guessed. So one of the study sites that Mark set up was this site in New Zealand, this Franz Josef Glacier. And there's been repeated glacial advances from about 120,000 years ago through the present. And this is a picture of it today. And the position of the glacier has changed in the last 150 years, where it's gradually thinned and retreated with some minor advances. But what happens is the glacier moves, it deposits soil at different ages throughout the landscape. And so the glacier created soils at different ages, and this allowed us to do pyrosequencing of the bacterial communities in the soil, so using the RNA gene libraries to look at the composition of the communities. And we had soils from 60 years of age to 120,000 years of age. And at that time, the common thing was to do is we take all these sequences we get and we combine them, the sequences with 97% similarity to each other, and these would be called OTUs. And so the idea is that these are closely related, and so presumably they share a lot of phenotypic properties and similar distribution. So at the Franz Josef Glacier, out of 90,000 reads, we found about 4,700 OTUs. And while probably some of these are sequencing artifacts, probably most of them are real. Now, I just want to point out that I think we did 5 or 6 of these chronos sequences. And so the results, I'm presenting this result not because it was typical of what we found in the other ones. And so when you have the OTUs, one thing you do is you make a big table, and this is the 25 top most abundant OTUs we found. And one thing that's interesting, if we just look at this column, this is the total, there were 7,000 of one OTU out of 9,000 sequences. So out of 90,000 sequences. So about 8% were were 1 OTU, and this was something I don't think we had anticipated. You know, I would have expected that there were a lot of very small OTUs. However, we found in these soils that there were OTUs that were, 8%, and I think down to, around 900 here. So this would be about 1%. So these were OTUs that were all very abundant and easy to find and well distributed across the entire chronosequence. Now, most of these you couldn't actually identify, so here we just have a taxonomic group, but a lot of their actinobacteria, alpha proteobacteria, and which are common soil organisms. Here's some planktomycetes. There's some that we weren't able to identify, and presumably are, some other phylum that's common to soil. So this is nice, but what really is an OTU? And in other experiments, we've sequenced some of these OTUs with Sanger sequencing, and we've shown that, first of all, they're not one sequence. They're a collection of closely related sequences. And so there's a lot of microdiversity, and the diversity is very likely real. So when we use Sanger sequencing, where we're sequencing longer pieces of DNA, and where we curate the sequences by hand, we're convinced that this diversity is real. And then at 97% sequence similarity, an OTU is really just a cluster of related species. So it's not a single, it's not like a single species of a plant or an animal. And most of these soil OTUs are not related to commonly cultured bacteria, so we don't really know very much about them. And to emphasize this point about what an OTU is, I just want to go over briefly the definition of a species of prokaryote. So generally, prokaryotes are classified in the same species when they have about 97% DNA hybridization, or the melting temperature of their hybrids is greater than or is less than 5. The change in the melting temperature is less than 5 degrees. And this turns out to agree well with what we see now with genomic sequencing. So if you compare this to numerical taxonomy, which is a comparison of the phenotypic properties, everything above 70% here would be in a species, but this includes a large amount of phenotypic similarity as measured by numerical taxonomy. So a prokaryotic species is not anywhere nearly as closely related as what we see in plants and animals. And in terms of ribosomal RNA sequence similarity, a species is at 97%, it's actually above, it's probably 99.5% or maybe 99.1%. It's arguable. Now, we know intuitively this is true because you've seen it all anecdotally many times. So like here's an example of a group of bacteria that by the definition I just gave would be one species. So Bacillus cereus, Anthracis, and thuringiensis. They're common soil bacteria. They all form endospores as resting cells. Bacillus cereus causes food poisoning in milk. Bacillus anthracis produces the deadly anthrax toxin. And Bacillus thuringiensis produces the insecticidal protein, and you can go buy it in the hardware store. But these are all one species of a bacillus, probably Bacillus cereus, and by the definition we really use. And so these are examples of organisms which are really very different from each other, have very different properties, but have greater than 99% 60ness sequence similarity. Likewise, another example is E. coli and Shigella. They're both common enteric commensals, but one is a severe pathogen. And so these are organisms that have very different effects, and but are very closely related genetically. So we can imagine that within any single OTU, there are a group of organisms which are sort of similar to each other, but could be having very different effects on what they're doing, and say their interactions with plants or insects or invertebrates. Now, what if we apply this criteria, the species definition for prokaryotes to eukaryotes? So this is change in melting temperature for primates, and most primates would belong to the same species. So chimpanzees, humans, and gorillas, if they were prokaryotes, they'd all be in the same species. And I think that makes sense. That's believable to me. So now let's go back to our distribution of OTUs across soils in different environments. And so we can say these OTUs are fairly large groups of organisms. They're related to each other, but they're not necessarily functionally identical. And the most abundant 25 probably represents somewhere 20 to 30% of the total prokaryotes in the soil. Now we can also look at the distribution, and we see some OTUs where the distribution seems to be constant, whether in soil that's 60 years old or 120,000 years old. And then we also see OTUs where they seem to be becoming abundant in the older soil. So each column is normalized to 10,000 sequences. So we're making a fair comparison. So this OTU3, for instance, was nearly absent in the very early soils, but became one of the most abundant in the older soils. And we see all types of variation. This guy became abundant after 5,000 years. This guy became abundant after 60,000 years. And then there's some that are only abundant in the Middle Ages, like this guy, or some that are only abundant in the younger soil. So the question is does this really mean anything? What can we say about this? And it's easy to look at these changes in distributions of the OTUs, but really there's so little known about them that the significance of these changes is really difficult to understand. But, in large part because of the metadata we had collected with these samples, we could conclude that the populations were changing probably in response to changes in the soil chemistry and pH and nutrients, and they probably weren't well correlated to the changes in plants at this particular site. Now, we can step back to try to get a better idea of the total diversity in these soil communities. And so in this case, I'd like to talk about the diversity at a higher tax as well. And so this is a study from crop, pasture, and forest soil in Georgia, and we collected about 3,700 well-curated Sanger sequences. And this is a rarefication curve calculated at different levels, OTUs at different amount of diversity. So the top one is a diversity of 0.01 or 1% diversity, and then 0.05 would be at about 95% similarity. Now, 0.05 is about the cutoff level for bacterial genera. So when the members of the same genus generally have a 16S ribosomal sequence similarity of greater than 95%. And there's some exceptions I'll talk about. But in this sample of 3,000 sequences in soil from very closely related, we found about 800 genus level taxa were observed. And so the question is what can we say about this, and what does it really mean? So this is some recent work from a paper in 2014 looking at the accumulation of ribosomal RNA sequences in the SILVA database. And so here we have over the last 30 years or 20 years, we've had an accumulation of sequences added to the databases, especially with the advent of next-gen sequencing. And in the 1990s, there were maybe 1,000 sequences were added each year representing new genera. And then in the last 6 years, about 10,000 sequences added that were representing new prokaryotic genera. So that means there was a sequence added that had less than 95% sequence similarity to any pre-existing sequence in the database. So at this point, there's tens of millions of sequences added each year, and about in the last year, there were about, say, 1.4 times 10 to the 4th sequences with less than 95% sequence similarity to any other sequence in the database. And so this would be the discovery rate of new genera on Earth. And this is, since this is a database, it's accepting sequences from all over, from all different biotypes and habitats, and many thousands of different investigators. However, if you look at the number of entries, the percent of genera that are found in new entries, you see almost a linear correlation with time. And so this is the percent of new entries heading straight to 0. So the implication is that within the near future, or actually this was done in 2014, so probably by today, there's a good chance that most of the genera on Earth will have been discovered, at least the ribosomal RNA sequence will have been discovered by this method. And this tends to show the rarefaction curve. So this is as you're adding new sequences, the number of genera that are found increases, and it's starting to plateau off. And it's estimated from this there's probably about 100,000 different genera on Earth. Now, maybe it's 200,000, but the difference isn't really, at this point, is almost immaterial. Now, we had about 1,000 genera just in our little sample in Georgia soil, and we've got about 100,000 genera on Earth. So we've got, 1% of all the genera on Earth are in my plot of, this little plot of soil in Georgia. Now, I don't know what it means, but it seems like it should mean something, right? So what is the genus? And so now we'll go back to comparing the ribosomal RNA sequence similarity from bacteria into symbionts of different insects. And so in the insects, there's a fossil record, and it's been shown that the bacteria and the insects co-evolve. So you can date the divergence of the bacteria from the date of the age of the insects. So these are aphids, cockroaches, tsetse fly. And so this is the general 16S sequence divergence in terms of millions of years based upon the insect evolution. So at 0.05 would be the genus cutoff, and so this would be about 175 million years old. So this suggests that a bacterial genus is about 175 million years old, give or take 50,000 years. So just for simplicity, let's say 200 million years old, excuse me. So at this rate, the age of most bacterial genera would be about 200 million years. Now, this is a group of related alpha proteobacteria, and this is a group of bacteria that, by the definition I just used, would all be in the same genus. So the Agro Bacterium, Rhizobium, Cynorhizobium. And so would this make sense as a genus? Well, Agro Bacterium causes crown gall, and so it infects one part of the plant and causes a complex symbiosis, and Rhizobium infects another part of the plant called bacteroids. So this might be a genus that's adapted to become symbionts or parasites of plants. And so that makes sense that this is a closely related group of organisms, but they're very different. But they're one of this, there's 100,000 of these groups on Earth with about this amount of similarity and this amount of difference. So what if we apply the same criteria to eukaryotes? Well, this is a wombat, a pig, horse, mouse, rat, human, and they would all be in the same genus. So the mammals and the marsupials would all be classified together if they were prokaryotes. They have about the same amount of genetic difference as we find in a genus of prokaryotes. And in fact, if they were prokaryotes, they're all heterotrophs, aerobes, they're oxytrophic for amino acids and vitamins, and they stain gram-negative. So if we use the same criteria that we use to group prokaryotes, they fit perfectly as a nice bacterial genus. And to make it sweeter, they're about 175 million years old. So Now, it's true that the mammals, the genetic diversity of mammals is fairly narrow compared to some other eukaryotic groups. But the reason to bring this up is it just emphasizes, and we say, this is the genus Escherichia, this is the genus Bacillus. We're not talking about, pine trees here. We're talking about like big chunks of biodiversity, really very different organisms, functionally, potential to do extremely different things. And I think that's a real important point. So now, can we answer some basic questions about the soil microbiome? So who is there? What are the communities like? Do they change? Does the vegetation affect the composition? And to do this more precisely, we worked with Steve Rathbone and Ming-Hung Kao, who's now at the University of Arizona, to develop a new statistical tool called K-Shove to help us evaluate the relatedness between libraries. And this is based upon something called Ripley's K-function, which is used for spatial point analysis. So Rathbone studies topology, and his basic question, if you know the distribution of A and B in space, how do you tell if the distributions are the same or not? And this turns out to be very similar to the situation we have in these RNA gene libraries. It's just we assume that the space is multidimensional. And so this is an example of, we have an intra-K function which measures the diversity in the library. And so it reflects both the number of O2Us and the abundances. So if you look down here, here's a hypothetical library. These are the distances between the four members, and this would be the intra-K function. And so the idea is that at point 1, there are no members of the library that have whose distance is less than 0.1 or less. So this is 0. At point 2, there are 30% of the comparisons are 0.2 or less, and then at 0.4, which is all of them, they're all at 0.4 or less. So that describes a curve here, and that's the intra-K function. And we can also look at this area here and say the area is a measure of the total diversity. Now, this is a different K function, because this branch is more slimmer and this branch is more different. The curve is different from here, but the area here is the same. So you can get different, if you use a single metric for diversity, you can get the same number doesn't necessarily mean they're the same, but it means that the total overall similarity is in some sense the same. And then likewise, here the diversity is greater between the two branches. This number is larger. Here the diversity is greater within the branches. The number is larger again, and now everything, it's larger again. So now it's 0.53 versus 0.5 versus 0.47. So this reflects the total diversity within each library. Now the cross-K function does something very similar, except now you compare one library to the other. And so in this case, these are the IK functions of the two libraries with the solid and dash. They're both the same. The cross-K function is 0. Now the libraries are different. And so it's the difference between the intra-K functions and the cross-K function that gives you this number here. And then as they become more disalike, more different, this number grows. And it turns out that this is more sensitive to change than unifrac, which is the other popular way to do this. And in library mixing experiments, which I'll show in a little bit, it's possible to show that the differences were approximately linear or monotonic with the differences in the libraries. And so this is some real data. So this is seawater and marsh sediment and the cross-K functions up here. And I'm going to try an experiment here. And then these are just UPGMA plots of what you see from the different types of comparisons. So here's the cross-K function. You have two soil libraries, CS, crop soil and forest soil. against the marsh sediment. The soils are closer together, like as they should be. The marsh sediments are more different, and then this is seawater, which is very different from all three of them. Now, if you look at the unifrac comparison, the topology, the branching order is the same, but the distances make it look like the soils are almost as different from each other as they are from the marsh sediment and seawater, which we know is not true. And the Wade unifrac is very similar. So the cross-K function then provides, we think, a more intuitive distance measure than unifrac. However, I will say it's not an exact measure at all. To look at the sensitivity, we use power analysis, and in this indicated that the measure is sensitive even for very small libraries, and the distance measure increases monotonically. So in a power analysis, what you do is you take two libraries and then you mix them. So first, if you take the unmixed libraries and say this might be soil and marsh sediments, when they're unmixed and you determine the K function, they're significantly different. They're 95% chance of being significantly different nearly 100% of the time. If you mix them 50-50, then they're the chance of being 95% different 5% of the time, or what you'd expect randomly. And then in a power analysis, what you do is you just increase the mixing proportion of the different libraries. So like when you get to 70%, if you have 100 sequence libraries, you have 30 sequences, say, from A, 70 sequences from B, and then in the other library is 30 sequences from B and 70 sequences from A. This means that both libraries, 60% of the sequences are from the same libraries, and only 40% are from either A or B, is what's different. And so when you do this with K-shove, with 100 sequences in each library, they're chance of being 95% different 100% of the time, or nearly 100% of the time. And even when you only have 50 members of each library, then they're different about 95% of the time. If you do the same thing with unifrac, with a 50 member library, they're different about 20% of the time, and lip shove is close to 40% of the time. So these two measures, which have both been in the literature now for over a decade, are just not nearly as sensitive as this method. Now the other thing is the distance really a linear distance? And so if you compare two very different libraries, so this is water and sediment here, and as the mixing proportion increases, you see it linearly increases to what you get the distance when the libraries are unmixed, except for a little bit down here. When you compare sediment and soil, where the libraries they're different, but the diversity within each library is similar, you see a monotonic increase in the distance, but it's not at all linear. So you have to bear that in mind when you're trying to interpret this. So K-shove also works for Illumina data. Most of this work was done before Illumina was widely used. And in the studies we did, we used 100 sequences per sample were sufficient to resolve closely related libraries. And so this high sensitivity allows you to analyze individual OTUs from large data sets, like that OTU, the first OTU in our soil study could have been analyzed, just that single OTU could have been analyzed for changes in the composition in different samples. But you can also use small samples that might be generated by multiplexing systems, which might have 1,000 different libraries in one sequencing run. And this might be important if most of your diversity is intersample variation and not, and the individual complexity of the samples are fairly simple. Now, this is looking at a K-shove comparison of RNA gene libraries from crop, pasture, and forest soils, and this is based in total on 12,000 Sanger sequences from Georgia, Kansas, and Michigan. And so I'll go through and explain what each of these are. So in Georgia, we went to the USDA J.P. Campbell Center. In Michigan, it was the Catalog Biological Station, and the Kansa Prairie in Kansas. And so the Georgia samples are all here, and you see that they're, these are kind of all are related, similar to each other, suggesting there is some geographical sense to the distribution, but not a lot. This is the cropped soil that cropped Georgia, and this is a soil that hadn't been disturbed since the Civil War. So it's a kind of a forest that it's been lumbered, but it hasn't been plowed. If you look at where the Michigan samples are, though, many are very close to this set of Georgia samples. This conifer forest, it's a plantation, is fairly different, and that seems to be a common property of these acidic soils. They're different from the others. And then we have the Kansas or the less. And so this is from a prairie site, and these all cluster together. So one thing, though, is if you wanted to say they're geographical distinctions, maybe, but most of the comparisons are very close to each other. So it's not geographically distributed. If you look, here's the Georgia, here's the cropped. These are two pasture plots, and then this is the forest. And so management seems to play a big factor here. If we look at Michigan, here's the crop there. This is a pasture. This is a natural pasture, though. It's been a meadow as long as anybody can remember. And then the forest is right next to it, and this is a forest that hasn't been changed much. Now we can look at transitions. So this is a case of the cropped was allowed to go to successional forest about 55 years ago, and then we presume this forest is heading into a natural forest. So there was a big transition in the community here. And now we also could look at a transition. This is, I think, about 12 years. This is the crop was just allowed to go into sort of grassland, and we think it's headed toward the meadow. So we do see transitions between the communities, but they take a long period of time. So even 50 years for things to happen. Now here's we're looking at Kanza. This is the cropped, and this is prairie, which would be our comparison to pasture. They're very different. These are all soils which have been allowed to become restored prairies. So this one is this is about 8 years, the transition here, but then you go back another 20 years, and then you're down here to 78, and then eventually you imagine it'll get back to the original prairie. So we see that the transitional states aren't necessarily between the starting condition and the end condition, but sometimes they seem to move around. And so can we answer some of these basic questions? So who is there? We can say who is there, but we don't really know what who means, So, and I would say, in fact, probably the biggest question in microbiology now is we really don't have any idea what most of the microbes are doing on Earth. They're there, they're abundant, they're different, so they must be doing different things, but we don't really know what they're doing. So the day in the life of a bacterium is a very serious scientific question, I think. For the structure of the communities, we can recognize communities with the same structure and describe them. We know that the soil community, the response to change is very slow, and we can identify transitional communities. And at least for the bulk soil, we think vegetation certainly has an effect. It's probably not the major effect. It's one of many effects. So I'm going to give some conclusions, and I'm going to give a very brief second talk, just because I know everybody's getting tired and they want something new to think about. So I want to leave you with something new. But first, I just want to restate the conclusions from here. So the soil microbiome is the largest microbial community that we come into daily contact. The community is extraordinarily diverse and comprised of many poorly described prokaryotes. The communities are relatively stable and respond slowly to change. And then soil microbiology is one of the great frontiers of microbiology. And now I want to just diverge a little bit and give sort of a grand vision. Where does this fit into the rest of life? And I kind of developed this notion that the motion of the planets of the solar system can be described by Newton's law of gravitation, which is written here. Force is equal to the gravitational constant times the mass of the two bodies divided by the square of the radius. And we say, well, what would the description of prokaryotic life on Earth look like? I mean, is it going to be an algebraic expression like this? And I say no, but I say it actually is going to look something like this. It'll have three major components. One is just the individual cell is described by the genome sequence. And in theory, the entire potential of the cell is in the genome. We just don't know how to read it. But we're making tremendous progress there. And so as we learn to read the sequence of the cell, of the genome, we'll learn what the cell really is. And in theory, we'll learn its entire potential. And the other major components, we have to know the relationships between the cells as described by their phylogeny. And to some extent, this is this argument, what is a species? How diverse is a species? What is an OTU? What is a genus? And I think, you know, we're making progress. We've got a long ways to go. And the third thing is a lot of what I was just talking about. How do you look at the distribution of cells in the environment? So I think if you know where the cells, where and when the cells are, their relationships between them, and you know how they function, you actually have a pretty decent description of life on Earth. And the solution to this equation is finite, and this means that it will actually, there is an answer, and someone will discover it. It probably won't be me, and it'll probably occur in the next couple of decades. And it's certainly not going to be one person, because these are really big questions, and there's a lot of information that needs to be collected, and we need to figure out ways to put the information together, and so we can share it and interpret it. And I just want to reiterate that a genome is probably fundamental to the breakthroughs in prokaryotic biology. So if you have a genome sequence, you can predict the physiology and ecological properties, determine metabolic properties, growth conditions, oxygen resistance mechanisms, toxins, and phenotype. There are biotechnological applications, novel enzymes, antibiotic biosynthesis, degradation of pollutants. We'll get an understanding of evolutionary processes, evolution of pathways, pangenomes, transmission of mobile elements, horizontal gene transfer, and then understand phylogeny and systematics. Now I bring this up because I'm participating in something called the Genome Encyclopedia Bacteria and Archaea 3, and this is a DOE Joint Genomes Institute project to obtain draft genome sequences of type strains of prokaryotes associated with soil and plants, and their close relatives, as well as type strains from newly described species prior to publication. And so we also have a second project I've called GIBA 3+, where we'll sequence pangenomes of 5 to 10 strains from the same species from soil or plant-associated prokaryotes. And the University of Georgia serves as a portal for this, so the genomes, genomic DNA is sent to us, and then we combine it and send it off to JGI for sequencing. And so if you'd like to email, to contribute, just talk to me or email me. And I say the University of Georgia, and that really is me, since you email me and I say sure, or I say that's stupid. But generally the idea is, we believe, collect as many genome sequences as possible. These sequences allow improvements of interpretation of metagenomic experiments from soil, as well as all the other things we said. And so we're really hoping to get good response in this. And we have 1,000 sequences for each project, and we're about halfway through the GIBA 3, and we just started GIBA plus the pangenomes. Well, actually, this is the first public announcement, so today. So if you have strains that you'd like to contribute, just let me know. And so the research here was supported by NSF and USDA, and thank you very much. Yes. Yes.
Well, with the plot, why do you have a definition of OTU? Perhaps.
Say what?
Why do you have a definition of OTU and you are showing the diversity, the plot, 0.01, 0.05. So if you could go back to the plot,
Yeah.
probably obvious, but probably not.
No, it's probably something that it's probably common in the ecology literature that we just don't.
Only for.
I know, but I'm jammed here. Here we go. There should have been an easier way to do this. This.
Here. So the question is that within the genus, there are probably strong selective pressure, so they are not going to go far from each other and evolve right around that. So if there's a big, selective pressure, they are not going to go far from each other and evolve. Keep the genus the same and adding few things. So you get a big diversity with a narrow definition. Whereas if you go too far, then they're not going to go far from each other. So it's probably selective pressure.
Well, so the question is why is there a genus and why is it that groups of 95% sequence similarity, 5% difference? And people are beginning to study this, and especially now that we have genomes, like we have a number of genera where we have good representation, like 20 or 30 genomes from the same genus, and you can form very robust, very good phylogenetic trees for the divergence of species within the genus and try to figure out what's going on. And I think it's I don't think every genus is going to be the same. I mean, some genera will probably be complete baloney. They'll probably it's just a coincidence, and you happen to grab a bunch of things and say, well, this is 95% sequence similarity, so we put them all in the same genus. And some genera will probably find some genera, there are probably very clear adaptations, like in that Rhizobia Agro Bacterium example, it seemed like that group of organisms was very clearly adapted to this plant symbiosis. They had different symbiosis, but that had to be a clear adaptation. And so to me, I look at it as more as a question. Once we start getting the information in, once we've looked at a couple of thousand genera, I think then we can draw real generalizations and have a really powerful impact on the way we think about this.
So related question is, if you see particular genus evolves in a particular environment quite a bit, are they getting more fit? That's why they're evolving that way through horizontal gene transfer.
So why don't we have just one genus? The most fit genus just take over the whole world. And I don't know. I have a theory that prokaryotes evolve very quickly, and because of their population sizes are so large, like E. coli is about 10 to the 20 individual cells, and that means every generation you probably have at least one case of a triple simultaneous mutation. And so you look at the gene space that could be reached by E. coli, it's enormous, because not only do you have all the sequence space, the gene space it occupies now, but then you have this huge mutation potential that it could reach within just a single generation. So if you say a genus is 200 million years old, it's done that, millions, hundreds of millions of times possibly. So the mutation space is huge. So I think, when a new environment is created, organisms adapt very quickly to fill it, and then within even just a few 10, 20 million years, which is nothing for these guys, 10 or 20 million years, they've fully colonized, fully occupied, fully explored that information space, and then they produce a stable population that maybe just cycles around, doesn't change. Now, is this true? I don't know. It's fun to think about, and I'm hoping these are the kind of issues that are going to come out. Our children are going to figure it out. So, and they're testable questions. We just vary at the beginning, trying to figure it out. And by this hypothesis, why is chlorophyll photosynthesis seem to have only evolved once? And you might say, well, maybe it required 5 or 6 simultaneous mutations, which probably only occurred once or twice in the history of life. So, that's one way I look at it.
So I was going to, I guess talk about this idea of the rare sequences, which, I guess there's debate about the importance of the rare sequences. I think I always think of soil as a special environment because it actually provides so many microenvironments that you think of there's this high potential for diversity.
Yeah.
But then when you look at marine samples, and I guess in some cases it's marine water, but in some cases it's actually the sediment, you're still seeing pretty high diversity there too, right?
Well,
or do you think the soil is relatively more similar to sediments in terms of a true maybe richness of types?
When we compared like the marine sediment, which these are mostly tidal marshes or they're coastal sediments, they were very similar to soil in terms of diversity. The compositions are different, but the diversity by number of measures are very similar.
But then the seawater.
Seawater is very, very much less diverse.
Okay.
It's dominated. You can get 20 to 30% of the sequences in one OTU. Now, I don't know about the long tails. So the tail may be really long in both cases, but I do think that everything I've seen suggests that the soil is much, much more diverse than seawater.
Well, I think it's one question that, getting back to what you were saying as far was that I always think of soil as a unique environment. If there was some way for us to go back, and sometimes when we study these chronosequences, we're trying to find sort of these pristine environments. And, a gram of soil, can have a surface area of several hundred meters squared. And then it can also have sequestered little pockets or maybe tens of thousands of bacteria can be separated from other bacteria. So you're kind of, to me, it's this potential at least for a lot of evolution to take place, a lot of divergence, potentially less mixing. But now that we've come along as humans, most of the space that we have now has been greatly mixed.
Yeah, but if you say, this was one of the things that I thought that I was surprised. I think that our data kind of disproves that in two ways. One is that the evolution of a prokaryotic group is in the order of millions of years, not hundreds of years. So, I don't know what, a genus we said was 200 million years old. So a species might be, I don't know, maybe 5 million years old, maybe 1 million years, but 1 to 10 million years old. So you're not going to see any significant evolution in sight in that particular soil. And I thought from our data, we kind of eliminated this idea. I thought when we started, under every rock was a unique environment, and so you'd see a different community. But especially in the forest soils, which were not disturbed, we saw that the community was fairly uniform.
And but as you admit, though, that the ability for like 16S to actually really look deeply and truly at the differences that represent. If you go back again to your Agro Bacterium example, you got two symbioses that are the same, but they do. One's a pathogen, one's a mutualist.
Yeah. One's
helping the plant, one's actually antagonistic to the plant. Very closely related, but doing very different things. So you can say even within that 0.03 or maybe 0.01 in some cases, we're not even coming close to describing the true diversity that's there.
Yeah, you're okay. That's right, and you're right. We really should be looking like metagenomic data or looking at specific functional proteins would probably do a much better job in terms of functional diversity. But I think soil's got to be mixed. To explain our results, to my sense, it has to be mixed. If not maybe on the order of years, or maybe on the order of 100 years, but But
Much quicker, much smaller in terms of time than the evolutionary perspective.
right,
Yeah.
and I can't prove it, and we haven't really tried to, but
I guess when I think of soils, I look at even some of these chrono sequences, like we haven't looked at any that are that old, but why they have chrono sequences with soils that are millions of years old, for example. And then you start getting to the potential for an organism to be sequestered away for, potentially, millions of years. And I don't know, it's all hypothetical, but it's just fun to think about, the potential for that contributing to the diversity that's found, a soil environment per se, yeah.
okay.
Can I ask one quick question related is, to promote horizontal gene transfer, how much is the planktonic movement inside the soil? You are sessile, then you start moving, you go somewhere else. So you're talking about mixing inside the soil. Is it moved by some sort of a temporal planktonic movement inside the soil?
So is there a planktonic movement of prokaryotes in soil? I don't know.
Has anybody measured?
What?
Has anybody measured?
Has anybody measured it? And you guys?
Was the life as a sessile organism then moving to planktonic and becoming sessile again?
So you mean something actually the plant root actually moves it, mobilizes.
The organism moving within the microenvironment, soil.
Yeah, no, I think there is evidence that it does, but
I mean, insects turn over, earthworms turn over soil on some basis.
Yeah.
But I don't know what the rates are, and I don't know if their bacteria motility would take them, how far they could go.
So what people call about recharge in the soil, water level payments, because things are moving around with the water too, right? Seasonal. All the microheterogeneity is kind of moved away by the movement itself, not necessarily motility.
Yeah, those would be good things to. I think they're interesting questions anyway. All right. Well, thank you.
Questions? Okay. Thanks, Barney.
Yeah.